everybody, and welcome to today's webinar. This is the fourth webinar in our series, Bringing Data to Life, Data Management for the Biomolecular Sciences. My name is Melissa Burke, and I'm a Scientific Training Officer in the training team, and I will be your host for today. So with that, it's my pleasure to introduce today's webinar, which is about giving data context, structure, and meaning. And our speaker for today is Sira Santofigio. Just give me a moment and I'll open her biography. So Sira works in Elixir technical team to support the scientific research across Elixir's 21 member states. Prior to joining Elixir in March 2018, she had been part of the Samples, Phenotypes and Ontologies team, also known as SPOT at EMBL-EBI, as an ontologist. Her expertise lies in the area of biomedical big data integration by the implementation of ontologies, linked data, and semantic web technologies. She is a developer of the cell line ontology, the ontology of adverse events, and the experimental factor ontology, also known as EFO. She is also an active member of the OBO Foundry community. Prior to joining the EMBL-EBI, Sira received her PhD in bioinformatics from the University of Michigan, Ann Arbor, and an ORISE postdoctoral fellowship from the US FDA's Office of Clinical Pharmacology, Office of Translational Sciences. So I'll hand over to you now, Sira, and I hope you enjoy the webinar.
Thank you, Melissa. So I'm going to turn my webcam on. Now you have a pretty good idea of what I look like. Giving data context, structure, and meaning. So as Melissa mentioned, I am now at Elixir as the interoperability platform coordinator. And before we go there, before asking, so why on earth would you want an ontologist at Elixir? Let me give you some background on what Elixir is, because I get asked a lot on what we do. What is Elixir and what does Elixir do? So Elixir's mission is to operate a sustainable European infrastructure of biological information to support life science research and its translation to society, the bio-industries, environment, and medicine. So that's a mouthful. So basically, we try to, our strategy is to coordinate across various different disciplines from the perspective of software, hardware, data resources, training, and then the interoperability, because you will see in the next few slides that looking at the various perspective and the diversity of data and resources that we see today across the different domains here, environment, bioindustries, agriculture, and medicine here. There are certain needs arising to coordinate across the different data with the standards and the interoperability. Elixir's strategy is connecting the national bioinformatics centers together, so we work at the national level. So as Melissa mentioned, we have 21 member states, including EMBL-EBI, and we do have Elixir observer states right now, which are Greece and Cyprus, who recently joined us in the past few weeks. And we span across Europe, as you see here, is the majority here. So we try to build an ecosystem for the open data innovative in Europe. So that's what Elixir is trying to do by building the portfolio of the CDR, the core data resources that are also listed on the Elixir website. We also try to identify the scientific data resources because they are the critical infrastructure and in the knowledge economy. By combining a lot of data together and identifying the sustainable model for the research infrastructure, we can save some resources, money, time, and other things. We also have the Elixir Innovation and SME programs that is comprised of more than 350 companies across our member states to support open data business models and opportunities. So there are some examples here, and we do publish the reports annually. So now, how do we connect all the national infrastructures? There are some examples here that, for example, in human data, we have beacons and local EGA projects that federate the sharing of the human data and the data discovery and query services. We also look at plant data. So in this example here would be the standards and services for the plant phenotyping data. and we also accommodated global collaborations for standards infrastructures. So in a nutshell, this is probably where I interject in as the interoperability platform coordinator. So as the interoperability platform coordinator, what do we do? Because we have already shown you the various types of data in different locations, and we try to federate across multiple international and national levels. We have learned that as of today, for example, data is distributed. They no longer reside in one location anymore. So they reside in different countries. They look at different bits of the data in the same domain, so the data in the different domains. So one of the issue here to resolve this challenge of the data distribution is that we have to make data findable and accessible. How do we do that? We'll be discussing that next. Also, to analyze, to understand, to answer a biological questions these days, the questions gets more complex and more complex each day. And so data to support the finding of the answer of the complex question coming from different formats and from different resources. So data are also heterogeneous. They come in from different standards, different formats, different content. So we need to identify the standards that walk across between these different formats, and that to identify the interoperability of the different data sets. At the same time, with the technology that is evolving, data are also changing. It doesn't mean that data that we discover or generate today, if it looks different from the data that we will see 10 years from now, it means that today's data are going to be wrong. It's just that technologies are changing. We can see different DNA profiles at the different resolutions now, for example. So we have to address the issue of reusability and reproducibility of data and the research itself. So together, this is called the FAIR principles: findable, accessible, interoperable, and reusable. And that is the heart of the interoperability platform, pretty much anywhere, not just at Elixir. So for what I've just mentioned here, data FAIRification is the new buzzword. Data FAIRification to support linked data via semantic web. So within the half-hour time that I have, I will try to make this clear. So how do we achieve interoperability in a nutshell? When we look at data connections, if you look at these different colors in the diagram here, you can think of the data that come in from the same resources or the same domain. So if we see the blue domain, the orange domain, the yellow domain, and the green domain, some of these they have the directionality, they have the hierarchy. Some data can be made into classification of hierarchy. Some other data may be just a floating dots in the pink circles that you see here, but you can build some loose coupling or some loose connection to the different hierarchical data components. So from the technical perspective, if you look at the data from the different domains, each knowledge concept is each node, each circle thing. To make data fair, each node or each circle here will need a unique identifier. It has a set of incoming or outgoing edges trying to define the directionality. So for example, in a normal classification, you can have a child of a parent node defined as it is a subclass of the parent node, and that is the directionality that some data can contain. Each node, each circle here will have a different properties. It will become clear in the next few slides. So edges in the diagram here, you can also see that, as I mentioned, you can build the connections. Some of them you can have a very well-defined connections, and you would see a solid arrow line here that also define the directionality and a very well-defined connections or relationship between each node. But for some that are just the floating nodes out there, you might need a different mechanism to define connections between these nodes and the connection from these nodes to some structure information. And this is at the top level how you would look at semantic web linking the defined data together. So how do we verify data? So the standards that we are using today. So today is the way that we defined connections that can be interpreted across the different geographical locations or different domains of research at all. So think about our good old website, World Wide Web. It was defined by the World Wide Web Consortium, the W3C here, so that if you type in a URL, http worldwideweb.something.something, regardless to the browsers that you are on, you can also go see the same content. So W3C are the consortium that define all these different protocols and standards to connect the internet together. We use the similar, we use the specification defined by the W3C consortium. And in a nutshell, the core principles of the semantic web linked data is that unique identifiers may be in the form of the URIs, Unified Resource Identifiers, DOI, Digital Object Identifiers, or some other forms of the W3C defined identifiers. We then use the good old HTTP protocol, the Hypertext Transfer Protocol, in addressing finding where it is, so locating the information about this specific concept that you identify by the standard identifiers. From this perspective, because we have the identifiers that are standards across all the different disciplines, because we follow the World Wide Web Consortium specifications, we can use the URI lookup services, such as those that are implemented on top of the RDF framework or Sparkle query to find the connections from one node out to different nodes. And this is how you would build this connection graph. I will have some example here. I know that it's a bit hard to understand from this diagram. So let me give you a specific example from our use case at Elixir. So this is the Elixir rare disease use cases that you want to look at the different context of how the rare diseases developed in the human body. In concept, you would want to connect the genotyped data all the way to phenotype data. But before you can connect genotyped, such as the DNA sequence or DNA profiles, to the clinical observations of the phenotype data, you actually would have to understand the different bits of it. So for example, you want to understand the genotype data all the way to phenotype data based on the context of rare disease. But when you look at that, the understanding of phenotype data, you also have to understand where in the biological body the phenotype occurs. So that you would need the understanding of the organism species, the organ parts, the anatomical locations, or even the cell types of it. So on this diagram, you might notice a few bits here that in this ecosystem of understanding mapping from genotype to phenotype in the context of rare disease, you actually have to look at the raw data, be it BAM file or FASTQ file, run it through some standard analysis pipeline. So those data would be stored in EGA database, for example, run it through pipeline, and then accommodate on this Rare Disease Connect platform. At the same time, other bits that come in from the clinical environment would be samples from the patients run through a platform called Phenotypes that annotates the data using the human phenotype ontology terms, the HPO terms here. I think you may have seen the activities of ontology curation with ontology last week, but there is a link from the series that you can go back and watch it, because I don't have time to go into details on that. So just from this example alone, you can already see the various type of data that would need to be linked together to understand the context of rare disease. So if I refer back to this diagram again, now in the core principles, each concept, be it cell type, organ, species, disease, or gene components, if all these various concepts have a standard identifiers, URIs, for example, then it can be linked. And if I take that apart, you would see the different bits from like, think of the blue dots as the body parts, think of the orange dot as the disease, think of the floating pink dot as maybe something else that would supplement this with new information, but it's not connected to anything else. So if I take that away and I present to you this picture of the disease ontology called orphanet having Li-Fraumeni syndrome, which is the complex syndrome that would lead to the genetic predisposition of the different cancers in your body. It is a structured data type that can be classified into a subclass of the parent term of inherited nervous system cancer. But then notice this pink floating dot. You might have a book on hereditary tumor syndromes in neuropathology with these digital object identifiers listed here. At the same time, from this knowledge of the pink dot, you also know that the inherited nervous system cancer can also be linked to central nervous system, which is yet a different ontology describing the anatomy of the body called Uberon. And that also has a subclass of relationship that is a solid, stable relationship all the way up to its parent term of nervous system. So now, walking this graph, you can already link Li-Fraumeni syndrome to nervous system, even though in the definition of the syndrome itself is just the predisposition to various type of cancers that is grouped as a disease syndrome. So this is just one small bit in the big diagram here. So if you think of the complex diagram before, you can also walk back from disease to phenotype to different cell types to everything in the same manner. And this is just one example of it. So I got this out of the web, just simple Google. There are so many projects out there today that use these concepts to link data up via the linked open data framework. So this example is the Yosemite project, and there is a link here that they try to understand similar biomedical and medicine research, linking general and internal medicine all the way to the molecular biology, to neurology, to the different technologies in the context of understanding the diseases. So in the bigger picture, these are some of the things that can already be achieved with the linked open data just by building the connections based on structured data and unstructured data. So I've just given you an example of things that can be done, but how do we achieve that? How do we make data findable, accessible, interoperable, and reusable? So the title of this talk is Putting Context, Structure, and Meaning into the Data, but from the practicality point of view, it's actually the reverse of it. So the title, taking the top-down zoom in from data context to data structure to data meaning. But from last week, if you were sitting in the last week presentation in this series, you may have heard that we start from raw data. Data are curated, meaning you assign the standard identifiers to it, be it ontology identifiers, digital object identifiers, what kind of any kind of the standard identifiers to that. So that's called data curation. Data are curated, data are given label and meanings. After they are given labels and meaning, then we move on to the structure. Data are classified into different bins. So the data can be human cell types, can be different parts of the fish. If we look at the plant, it can also be classification of the plants. If you look at the animals as a whole, you may also look at the animal kingdom, for example. After data are given structure, then you ask which context are we looking at these data, because one concept, one knowledge concept can also be seen from different angles. By defining what question are we asking, we can try to limit the scope of how we define and give structure and meaning to the data. So that's the context of it. What questions are we asking? So in this example, it might be looking at the cancer progression and prognosis. What environmental factors might affect the cancer progression in your body? And then after that, because modern science, we already know that science knowledge is changing. Knowledge is a dynamic discovery. There will always have to be a reality check and recheck on what we know today. If it doesn't, what we knew yesterday, if it doesn't hold true today, we go back to this same circle again: data curation, data classification, giving the context, recheck. So these are the circle of the activities to make the data fair. So what are some of the examples of the tools and processes in each of the steps here? If you're sitting in this seminar, I get it that you have some interest in understanding the relationship between these 4 activities. So there are tools out there in assigning the identifiers. So we can use control vocabularies, which can be a flat list of things that you have an agreed upon definition. So think about dictionary, be it Longman, Merriam-Webster's. We can have that in the biomedical research as well, that an ear in an animal body is the organ part that would be receiving sound and translate that into meanings. But if you look at the different vocabulary set, for example, the plant dictionary, and a corn ear is definitely not an ear to listen to things. So that is the standard control vocabularies that you will have to select and based on the context of the question that you're asking. At the same time, you can have identifiers such as the digital object identifiers that I keep mentioning. Those are defined based on some agreed upon standard like the Blue3C consortium, for example. You can also have some other form of linkable identifiers implemented on your own end, and then you find a way that maps it all back to the controlled and standard linkable identifiers out there. I put ontologies between the 2 sides of data curation and data classification here, because for some data that can have structure, placing ontology identifiers at the data curation step also reduce the workload by eliminating the needs for having to identify the classification manually. This comes together at the same time, and that's why I put ontologies in between data curation and data classification. But what if your data are not structured? What if they're just Twitter feeds? What if there are just some publications out there, some books out there that you can't really define the relationship in the classification hierarchy as you would see in ontologies? You might use a knowledge graph out there. You might use some ad hoc classification that you have to build in the house, and then you have to think about the way to implement a link back into the standard framework. Then finding the answers after you have the data that are curated and classified, finding the answers to fit your context. What are some of the tools out there? We're starting hearing about the machine learning, artificial intelligence, integrative analysis. These are the platforms of finding the answer to the context that each of us is trying to understand. And then in reality check, I also put the machine learning and integrative analysis here, because sometimes when you run an analysis or you run your machine learning pipeline, you might also identify some new knowledge that might have contradicted the old knowledge or just some brand new knowledge at all. So in reality check step, you can have the community and the curator task force. You can also might find it via the integrative analysis or machine learning. So these look a bit messy, but these are some of the available tools out there that that can already be placed on each of the oval here to help you build your your environment of the FAIR data. So for example, for ontologies, you have ontology lookup services at the EBI. You also have the OBO Foundry, which is an open biomedical ontology consortium. But what if you cannot use ontologies? You might as well try to see it with the Google Knowledge Graph or using some information from linkeddata.org. So these are the tools that are available out there today. So I summarize some of the links that you might find useful here. If you're not sure what ontology is, the first link is a link to a recording of a detailed ontology 101 step-by-step that I gave a few years ago at University of Michigan. I hope the knowledge still holds. And what we provide at Elixir. There's some ontology tool suite out there. There's OBO Foundry. And if you really want to implement something, I would recommend you look at the W3C specifications here. The slides will be made available. So after describing all the different things that already are in place today, one of the many possibilities once the data are given meaning, structure, and context would be, you see, from the heterogeneous data, after adding value to existing data by linking it and bringing together with the different common standards and understanding. This is already happening today. So another example here is understanding the drug development and research from the safety perspective, the drug safety perspective, because not only that we are trying to understand the disease progression, the disease relationship in the body, when you have another player in the picture of drug research and development, for example, a lot of times drug failed, not because it doesn't work. Drug fails because it's not safe to take. So medicine is just toxin in a mild dose. But understanding how people develop side effects actually need a lot of components. It seems to be such a straightforward question, but it's so complicated to answer this question because we need to understand why different people develop the adverse events in their body differently. So these are some of the framework that we've been involved in, that there are scientists who are trying to build the platform to combine the preclinical data, clinical data, physiochemical data, molecular data out there. And then because of the amount of data that we're looking at today, all these will have to be integrated and analyzed on the cloud and using the drug safety data labs such as Transmart, and then validate it through some other algorithm to understand the side effects. So one example here would be understanding how a cancer treatment can lead to a heart failure. What is the mechanism behind that? So on here, there are two pathways, completely different. One is the kinase domain in the body, and one is the drug pathway imatinib, which is the inhibitor of it. It actually splits down here along this cascade. One would result in cell death, the other would result in cell survival. But understanding why, after what is the interaction between the kinase domain and the imatinib as the drug, in example here, we actually might ask, okay, what are the endothelial cells, which are the cancer cell, and the cardiomyocytes, which are the muscle cells in the heart, have in common, so that we can understand why after the patient received the cancer treatment, of course, the cancer cells might get killed, but at the same time, you also killed the heart cell as well. This is very complicated to answer, but because if we curate all the data the way that we've been trying to describe in the past 10-20 minutes here, we can use text mining. So this is my unpublished work on using ontology assisted text mining on PubMed dataset to identify the markers and the molecular activities that might explain the cardiomyopathy in the cancer treatment patients. And then So the green ovals here are the new knowledge that we found from PubMed, from text mining and artificial intelligence along the machine learning of it, using ontologies tagged data. There's some data that are agreed upon from the previously known knowledge in the yellow boxes here, and then there's some human curated perspective in the pink circle here. So this is the big picture that we can already build from this. And because what we found here, we can also reverse this back to the ontology concepts that, okay, we have myocardium that can be linked to myocardial infarction as the adverse event from the adverse event ontology. So in this picture, it's just a small example of how concepts from the anatomy ontology in yellow boxes are linked to the adverse event phenotypes in the blue boxes here. And can this be explained by the processes in the gene ontology biological process? So in conclusion, I've shown you that biomedical research is global, and that's why we have Elixir, because we need to engage at the international level to drive global standards. And because of this global attempt, we also need standard for defining meanings and structures for the collaborative context here in all software, hardware, middleware, and the interoperability underware perspectives. So that leads to metadata standards, compatible clouds, data exchange protocols at the national clouds level. And these come back to the virtual research environments. If you recall the picture of the rare disease use case that I showed you, that is the virtual research environment, meaning we hide a lot of complexity behind the scene, but there is a massive mechanism behind the scene that are combining all these different data, because everyone likes simple interface. Just think about Google search box. If you go to google.com, you just see one search box, but as soon as you start typing something in that search box, you are already using ontology in one form or another, using Google Knowledge Graph, for example, to get all the result back in less than 1 second. It will never be achieved if there is no ontologies or the knowledge graph behind the scene. And with that, I hope I didn't take up too much of your time. And I think we stay pretty much very well within the time. And I am aware that I try to cram so much content in such a short time. So if there are a few questions that I can help answer here, I will.
Thanks very much, Sira. Indeed, we do have time for questions, and you can write them in the chat box. While you're doing that, just a reminder that the next webinar in this series will be next Thursday, and we'll be looking in more detail at tools that you can use both to make a data management plan and to help make your data fair. So I think some of the tools that Sira mentioned in her talk will be looked at in a bit more detail. Perhaps I can start with a question. So during this series, we've been talking about the selfish benefits of data management. I wondered what your thoughts were on what are the selfish benefits of annotating your data with an ontology as a researcher at an individual level.
So research gets more complicated, and as it gets more complicated, it gets more expensive. So by curating your data right with these FAIR principles, you might ask, why would you want to spend so much energy and so much resource to do that? But if you think about the amount of time and money that you put into curating your data compared to what you gain back from being able to connect your annotated, your curated data out there to the bigger set of knowledge out there, that is the selfish benefit of it that I cannot see just like immediately. But if you think at the national or international level, that is also, you're combining a much larger scale, looking at different layers of biology, and at the same time, it's not just, if I were to be putting on my entrepreneur hat on, it's not just for the benefit of the mankind. If somewhere along the line you identify, oh, some microbiome interactions, and that also leads to biofuel, you can be green and then you can make money. So that's also another benefit, the selfish benefit, that if I were the SME looking into this is what I want to do. So there's also another example from the Pisteria Alliance, which they actually bring together a lot of the pharma companies together, because doing this together is much cheaper. And that's why we start seeing a project that are public-private partnership. It's like open targets.
Yes.
Elixir, that's why we have both the academic technical side of story, and we also have the SME partners, so that because we understand that to drive the global effort forward, it cannot be done just in academic setting or just in the SME setting or within just the pharma, and that's why we start seeing a change of landscape. I would take Human Cell Atlas, for example. Why would Chan Zuckerberg Initiative, the Facebook people, look into this? Because there is, in my humble opinion, there is a trend change in that academic and commercial. They're not standalone entities anymore. There are more and more interactions as we see it.
So I guess annotating your data makes it more visible and then more usable, and you can
make it cheaper.
Yeah. Cheaper, you end up interacting with people more, and if you're going to be selfish about it, that can really benefit your career and what you can discover. Are there any other questions for Sira today? Okay, so there don't seem to be any more questions. Thanks very much for joining us today, and I hope to see you in another one of our webinars soon. Enjoy the rest of your day.
Thank you, everyone.